Thank you for filling the present form. For the next session, I would like to welcome Mr. Rifki Wahyu Kurnianto, Master of Engineering, as the moderator, who will guide the course of our discussion for our second class. Before moving to the lecture, I would like to present a brief profile of our moderator today. Mr. Rifki Wahyu Kurnianto, Master of Engineering, is an expert in biological wastewater treatment, biomaterial, and fermentative biorefinery. He is a part of Bioreactor Engineering Expertise Group in Department of Chemical Engineering, Universitas Gadjah Mada. He is also a PhD candidate in Delft University of Technology, Netherlands, with his research topic in biological wastewater treatment. Now, without further ado, Mr. Rifki Wahyu Kurnianto is welcome to take the lead of the session. The time is yours.
Okay. Thank you, Theo, for the time that was given to me. And hello, everyone. My name is Rifki Wahyu Kurnianto, and I will be the moderator for the second lecture. Now, joining us today as the second speaker is another amazing researcher who comes from Japan. He is a microbiologist who has more than 30 papers in peer-reviewed international journals. He also established Alchemic Lab Incorporation, which is an enterprise dealing with the R&D on biomaterials production from biomass resources. He is an expert with prolonged understanding on cutting-edge wastewater treatment technology and how to create the function of microorganisms. Today, he will tell us about the microbial ecosystem on thermophilic anaerobic digestion. Let's welcome Dr. Yoshiyuki Ueno.
Hello, can you hear me?
Okay, hello, Dr. Ueno. Long time no see. How are you today?
Yeah, very good. But I'd like to share my materials. Just a moment, please.
Okay, Dr. Ueno, it's so good to have you here. Thank you so much for joining us today.
Yes, please. Thank you. Just a moment.
It's so good to see you again. Okay, Dr. Ueno, are you ready to present your presentation?
Yes, please.
Okay, then to all the participants, I hope that you are ready to learn something new from this lecture. And not to prolong the introduction any longer, Dr. Ueno, you are welcome to explain your materials. Time is yours.
Yes.
Okay, we can see it clearly.
Can you see?
Yes. Thank you, Dr. Ueno.
I'm terribly sorry. My name is Ueno. This talk is delivered from Tokyo, Japan. And first of all, I'd like to express my sincere appreciation to the organizing committee of our summer course 2021 and this workshop in IBASC 2021. Everyone, can you hear me?
Yes, Dr. Ueno.
Okay, I continue. Okay, and I also appreciate the Chemical Engineering Department of University of Gadjah Mada for giving me an opportunity to have a seminar today. I'd like to enjoy this workshop and make good relationship with Asian and African students gathering to this web meeting. The title of my talk is Microbial Ecosystem on Thermophilic Anaerobic Digestion. Okay. How to move over this? Okay, this is my face. I am a microbiologist. I was born in Tokyo, Japan, and I have more than 30-year experience in R&D in the field of biomass utilization, microbial material production, wastewater treatment. And I have belonged to Marine Biotechnology Institute, Japan, the Tokyo University, Japan Industry Association, and the Kajima Technical Research Institute, Japan. The most of the talk today is what I have done in Kajima Technical Research Institute. As you know, Kajima is the biggest construction company in Japan. And now Kajima is deploying business concerning environmental technology outside Japan. And Alchemic Labo is a small enterprise I established last year. And now I am a representative in this small company. The main framework of our business is R&D on the development of microbes and functional proteins, the fermentative production of variables from biomass materials and waste, and the consulting work. The consulting work is focusing on the promotion of biomass energy, the construction of international network, and overseas relation. So, I'd like to start my talk with this question. Since I am a microbiologist, what is fermentation? So what is fermentation? You might have studied it in the lecture for the biochemistry, microbiology, or biotechnology in your university. The term fermentation can be utilized in two different major words. One is anaerobic respiration, in the technical term of biochemistry, which is biological energy metabolism under anaerobic condition. It's release electron via oxidation. And another one is the microbial production of materials. The fermentation is utilized, fermentation is utilized in various kinds of product formation and production, such as ethanol, lactic acid, acetone, butanol, amino acid, antibiotics, and also hydrogen methane composting. The ethanol and other chemicals such as lactic acid and amino acid are produced by pure culture strains of microorganisms such as bacteria, yeast, fungus, and isolated from natural environments. On the other hand, hydrogen and methane, today I talk, and the composting work by mixed culture, which is a complex organism or system. So my title today to this seminar is Microbial Ecosystem on Thermophilic Anaerobic Digestion. This is a story of fermentation technology. So my talk composes these 5 items. Most of them might be duplicate and overlap to the presentation by Professor Samir Khanna. But I'd like to focus on the microbiology in my presentation. First, I talk general outline of biogas production, then the principle of biogas production. After that, I move my talk from fundamentals to engineering. At the final, I introduce an experiment in order to enrich thermophilic mesonogenic microflora that effectively work for biogas production from biomass resources. The title of my lecture is Microbiology, but In this seminar, I'd like to explain not only microbiology, but also how the basic scientific knowledge can influence and contribute to the engineering. In addition to basic scientific explanations, I'd like to touch on how activities are carried out in the private company and the company or industry with social and economic consideration. The content of my lecture may be a little technical, so it may be difficult for the non-specialist and undergraduate student to understand some part. But feel free to ask me if you have a question or something unclear. We may have a time after my presentation. So first, I explained the general outline of biogas production. I start my talk with methane. The methane is emitting from all kinds of natural environment in this planet. Disposal site, paddy field, and also cow barns, as can be seen in this figure. Those are dealing as a burden since methane has 25 times higher potential greenhouse gas than carbon dioxide. Towards suppressing occurrence, researches to avoid the methane gas emission have been taken worldwide. But in this seminar, I'd like to focusing on the production of methane, because methane has 55 megajoule per kilograms of calorie potential. It can be utilized as an energetic material. So methane fermentation is defined as follows. Natural phenomenon. Methane fermentation is a natural phenomenon. So that means the reactions occurring in nature. Then the reactions producing methane from organic compound by microorganisms in anaerobic circumstances. And the methane fermentation plays important role on the geochemical carbon cycle in various scale of environment, including deep sea, sediment, paddy soil, wetland, and intestine of ruminant animal insect, and others. And methane fermentation is applied to the wastewater treatment as having more than 100-year history. The energy production from biomass has much attention in recent years. And this technology is applied for the reduction of excess sludge. So it is called as anaerobic digestion. So apparatus for anaerobic digester is installed worldwide. This figure shows the gradual increase in number of actual plant from the mid-70s onward. As of 2007, more than 2,000 full-scale installations are in operation. At this point, I'd like to show you the comparison of anaerobic and aerobic for wastewater treatment. This figure shows this. So, in thermodynamics, that gives free energy is a thermodynamic potential that can be used to calculate maximum reversible work that may in thermodynamics, free energy change is the thermodynamic potential I can show you which I'd like to focusing on needs in aerobic treatment. Organics convert to microbial biomass. This is called anabolism. On the other hand, anaerobic treatment, organic materials convert to methane. This is called catabolism. So, this anaerobic treatment means much excess sludge production. On the other hand, anaerobic treatment can recover the methane as a biogas. So this is a feature of anaerobic treatment. As a pros, less production of excess sludge. The saving energy since aeration not required. Producing methane, yes, and less odor emission from closed system. On the other hand, as a cons, long retention time due to slow growth rate of microorganisms. The sensitive to environmental factors such as temperature, pH, loading fluctuation, and effluent quality inferior to aerobic treatment. And it might be not effective to low strength wastewater. This is a commercial plant of biogas production process constructed by Kajima Corporation. This apparatus is installed in the shochu factory. Shochu is a spirit, Japanese spirit made from potatoes. The mixtures of solid waste and wastewater from the factory is being treated at 800 tons a day of capacity. The biogas generation from 16 bioreactors is used for cogeneration by boiler for drying process in the factory. You can see the cylindrical plant in this photograph, bioreactors, thermophilic digester. And this apparatus, this facilities is generating 4 gigawatt hour of electricity generation per year. You can see this facility if you came to Japan. I'm pleased to take you to this site if you request me. And this is the operation data of soju brewing factory published in the journal. At approximately 14 kilograms COD per day of organic loading rate of 0.1 to 0.18 D of dilution rate, which correspond to the 6 to 10 days of HLT. Stable performance has been observed showing more than 80% of COD reduction with 75 of BTS decomposition during 70 months. This approximately 6-year operation. 18,000 cubic meter per day of biogas is being continuous through this operation. This biogas production is still continued to present. The performance is stable even now. As of today, Kajima Corporation has installed 15 biogas production plants in Japan. not only shochu waste, but also garbage and other organic waste are being treated by thermophilic anaerobic digester installed by Kajima Corporation. This is a costly ground garbage from the restaurant. This is garbage from the restaurant. 20 kilograms of garbage in the bucket is put, 20 kilograms of garbage is put into the bucket, as can be seen in this photograph. It becomes substrate for methane gas. It makes 2.4 cubic meter of methane gas, corresponding to 16 kilowatt hour of electricity. But if you leave this garbage as is, nobody can collect methane gas from this. It will emitting just bad smell. So bioreactor is required to utilize microorganisms effectively. Next, I'd like to explain the principle of methanogenic reaction. So this is a summary of methanogenic fermentation. Methanogenic reaction is the microbial decomposition of organic compound to methane by microorganisms under anaerobic conditions. The combined chain reaction by complex microbial system, which is called microflora. The bucket brigade of metabolite, intermediate metabolite by different species of microorganisms. Each elemental reaction complies with thermodynamics and stoichiometry. For this is material flow and microorganisms in anaerobic digestion process. Biomass materials, which is substrate for methanogenesis, is organic polymers, such as carbohydrates, proteins, lipids. Those organic polymers are hydrolyzed by hydrolytic fermenting bacteria to low molecules. Then these hydrolyzed low molecules are fermented to the VFS, alcohols, and hydrogen and carbon dioxide. And the final formation of methane gas and carbon dioxide is coming from just hydrogen, carbon dioxide, formic acid, formate, and acetate. These methanogenesis are performed by mesanogens. Mesanogens is classified into the domain archaea. You have to remember, domain archaea contains methanogenic microorganism, which is prokaryote, but it's not bacteria. For instance, I can illustrate methanogenic reaction from carbohydrates. Overall reaction is this. 1 mole of carbohydrate is converted to each 3 moles of carbon dioxide and methane. But the reaction proceeds step by step like this. Carbohydrate is hydrolyzed to glucose. Then this glucose is fermented to acetate and carbon dioxide and hydrogen. Then this carbon dioxide and hydrogen is converted to methane gas by hydrogen-trophic methanogens. And also, acetate can be converted to carbon dioxide and methane by acetoplastic methanogenesis. This is a principle of methanogenic reaction. Then I'd like to explain the microbiology in methanogenic reaction. So as I mentioned before, the microflora is essential to understand the independent microbial reaction. I'd like to tell you about the term microflora. This is a crucial term to understand anaerobic digestion process to produce methane gas. The microflora is complex microbial system composed by several kinds of microorganisms, which is naturally occurred. And methanogenic microflora is comprised by anaerobes, not only anaerobes, but facultative anaerobes and even aerobes. Comprised by wide variety of microorganisms among domain bacteria and archaea. And methanogenic microflora contains a lot of hitherto unknown microorganisms. So far, 700 to 3,000 species of bacteria and 10 to 7 species of archaea have been detected in clone analysis. They are differed in their characteristics among substrate, growth rate, and the condition, etc. And cooperative actions for biogas production from waste. This is a torrent of bacteria and archaea in methanogenic microflora. In the methanogenic microflora contains the two domains of microorganisms, as I mentioned. Bacterial community structure differed in the main substrate. Main substrate means starting material. And the bacteria is widely diversified. And archaea, on the other hand, less diversified. Archaea is a microorganisms produce methane in the bioreactor. Just only 4 species, Methanocurials, Methanocyanobacter as hydrogen-trophic methanogen, and Methanoceta and Methanocalcina as acetoplastic methanogen. Only 4 species are present, mainly present. And also I can say that they are dominant methanogen in the industrial methanogenic bioreactor. And I'd like to explain about the syntrophic relationship between the microorganisms. Again, I show you this material flow among a lot of decomposition reaction in the anaerobic process. Hydrolysis and fermentative decomposition of glucose, amino acid, fatty acid are easy, since these reactions are exoergonomic reactions. If we put or leave organic compound such as garbage, fish, meat, vegetable, or something in somewhere, they are naturally decomposed to organic acid and produce carbon dioxide and also make bad smell. But under anaerobic condition, decomposition of these VFA, volatile fatty acids, and alcohol is endoergonomic reaction, which implying that it doesn't proceed under standard condition. And the methane formation reaction uses only hydrogen, carbon dioxide, and acetate as substrate, and these methanogenic reactions are exoergonomic, as shown in the left side of this figure. So this decomposition of organic acid is rate limiting step of anaerobic digestion. So I explain the mechanism of syntrophic decomposition of volatile fatty acids. This is an example of propionate decomposition. As shown in this equation 1, propionate is decomposed to acetate and hydrogen and carbon dioxide, but this reaction calculated to be thermodynamically positive. So 76 kilojoule per mole of free energy change. This is impossible reaction to proceed. But methanogen can help to proceed this reaction, because if methanogens consume hydrogen and acetate to produce methane, as shown in equation 2, so that means that chemical equilibrium of equation 1 to trend toward right side, since these acetate and hydrogen are removed, and the partial pressure of hydrogen lowered in the right side by equation 2. So totally, methanogenic reaction by these syntrophic reaction from propionate, which is the combination reaction of equation 1 and 2. Free energy change can be calculated to the negative value, minus 56.6 kilojoule of free energy change. So methane gas production reaction became possible. It is so-called interspecies hydrogen transfer. Bacteria and methanogen cooperate to decompose VFAs. Methanogens consume hydrogen to produce methane. Acetate can be formed from VFA by removing hydrogen by help by hydrogen consumers. This is a microscopic observation image. Methane production archaea is stained green. Bacteria is red. In symbiosis, they are taking position close proximity in order to remove or consume metabolite from others. From here, I talk about bioreactor engineering. So this figure shows the cultivation mode in bioreactor. If you cultivate the microorganism, maybe you use a tube or RMA flask, but that might be a batch culture shown in the left side. In the batch culture, microbial cells grow gradually as consuming substrate. But on the other hand, continuous cultivation using the bioreactor, we can operate the like this figure shown in right side. The cell concentration of microorganism can be maintained at the same concentration, and the production volume also can be maintained and standard steady state of operation. So, what is bioreactor? Bioreactor is a reactor employing synthesis and or decomposition by using biocatalysts such as microorganisms or enzymes. There are various types of bioreactors. The most popular bioreactor is CSTR, as can be seen in this figure, continuous stirred tank reactor. This bioreactor, so-called fermenter, is just equipped with cooling and incubation unit to control temperature and the stirrer to mix the inside the media, inside the bioreactor. But there are various kinds of bioreactor, such as fluidized bed, fixed bed, or packed bed, or membrane bioreactor. They are slightly modified from continuous flow stirred tank reactor, CSTR. My talk today, I'd like to introduce packed bed reactor. This is a photograph in the packed bed bioreactor in the laboratory. The packed bed reactor, in the packed bed reactor, the supporting media is installed in the reactor, shown in the middle picture. The microbial biofilm is developed on the surface of the medium. It enables to increase microbial biomass inside the bioreactor. It also avoids washout of microorganisms at a short retention time. Therefore, high performance digestion is expected. We are using carbon fiber textile, CFT, as a supporting media installed in the bioreactor to develop the biofilm on the surface. This is a video. Can you see just a moment? Sorry, it cannot move, but this is a bioreactor, and you can see in the packed bed inside the bioreactor, this cylindrical form of the materials packed bed. At this point, I'd like to briefly introduce about microbial community analysis for the student who are not familiar with microbiology. Molecular ecological analysis is based on the criteria of central dogma of microbiology or molecular biology, which is a study of DNA transcription to RNA, then synthesized protein. The molecule in the environmental sample, such as sludge, tell us, in case of DNA, who is there. If we analyze DNA inside the bioreactor, what kind of microorganisms is in the bioreactor we can analyze. Who is not there. What functional genes are there. But we cannot tell you who is active if we analyze only DNA. If we analyze RNA, who is active, who is expressing gene, we can understand. And in our case, DNA is extracted from the biofilm and wastewater. Then PCR amplified specific genes are analyzed using NGS. NGS is next generation sequencer. The data obtained are subject to identification of microorganisms present in the sample and the evaluation and the comparison. Finally, we can estimate bacterial community composition. So this picture shows the microbial analysis of the biofilm developed on the carbon fiber textile in our packed bed bioreactor. This bioreactor was operated and OLR of 120 kilograms COD. This is an amazing OLR, organic loading rate. And HRT of this experiment was less than 2 hours. And this bioreactor ran more than 180 days. And in this bioreactor experiment, 75% of COD removal with biogas production was confirmed in continuous operation.
Excuse me, Dr. Wino.
Hi.
You have 3 minutes left to sum up the presentation.
3 minutes. Okay.
Yeah, okay.
Sorry. This is a result of microbial community analysis. You can see on the biofilm, archaea, which means the mesanogens, are highly concentrated in the biofilm. Okay, I'm in hurry. So biogas production from our packed bed reactors, microorganisms, most of microorganisms are present in the biofilm, but in the planktonic phase, the hydrolytic acid fermentative bacteria are presented in the planktonic phase. Okay, next, how to apply these microbial information to reactor engineering. We combine the data from microbial analysis and apply to reactor engineering, such as reactor design, the circulation, mixing fluid, media configuration, and materials and amount of media. These combination enables efficient digestion and acceleration of reaction rate, and also overall economic performance. I skip. So, final topic is enrichment of methanogenic microflora. This figure shows the planning to operation for biogas plant. So before planning the biogas facility, we have to take engineering data in the laboratory. How much biogas can be produced from the factory or facility of the clients. Then we proceed to plant design and construction. But before starting at the facility, the microorganism seeding is required. So to obtain engineering data in the laboratory, waste biomass is necessary, and also seed thermophilic mesanogens are also necessary to obtain these kind of engineering data. There are also large amount of thermophilic mesanogens are required to start up these facilities. So preparation of the seed microflora. If we are in Japan, seeding microorganisms can be derived from the present working plant and incubated in new constructed facility. But in overseas, we cannot deliver microorganisms from Japan due to regulation and quarantine and biodiversity. So we have to.
Excuse me, Dr. Ueno. I'm really sorry, but I think the time is up.
Sorry.
It's okay. We can continue if the question is still not there yet. But first, let me ask the participant. If you have any question, you could send it to the Zoom chat feature, or you could raise your hand, and I will ask you to unmute your microphone and ask the question directly. Okay, participant, do you have any question to Dr. Ueno?
Hi.
Okay, you are welcome to ask questions to the speaker, to Dr. Ueno. Okay, if you still don't have any question, I would like to ask a question first, Dr. Ueno.
Yes, please.
We are already familiar with the PCR because during the pandemic we hear it frequently to identify whether someone is positive COVID or not. So what I want to ask you about the NGS system, will it also measure the dead bacteria or only the live one?
Excuse me, once more, please.
Okay, for the NGS analysis.
NGS analysis.
Yeah,
Okay.
the NGS analysis, will it only measure the live bacteria, or it will only measure, or will it measure all the bacteria, whether it is alive or dead already?
Yeah, it's depend on the primer you used. PCR primer, if you use the covers all microorganisms, including bacteria and archaea, we can identify bacteria and archaea at the same time. So we are using V3, V4 region universal, V3, V4 region of 16S ribosomal RNA gene of the bacteria and the archaea. So we can determine bacteria and archaea at the same time by NGS. Okay.
Okay, thank you for the answer. So for the other participant, do you have any question? Hello? Any other question from the participants? You can send it via Zoom chat, or you can raise your hand. Okay, if there is still no question, I think there is still a few minutes left before the Q&A session. Before the Q&A session is finished, I think you can. Could you please finish the presentation in the meantime?
Yeah. Thank you.
Okay. Thank you very much, Dr. Ueno.
Can I share?
Yes, please. I think we still have about 10 minutes left. 10 minutes left for
we have
the whole
10
session.
minutes?
Yeah, 10 minutes left.
I'm terribly sorry for inconvenience.
It's okay.
Actually, this is my first time to present my presentation in Zoom.
Okay, it's okay.
I usually use
Microsoft Teams.
yes.
Yeah.
Just a moment. It's taking time. Okay. Okay, just a moment. Okay. So obtain thermophilic microflora outside Japan. So we have prepared microbial inoculant on site. So if we deploy the technology in Southeast Asian country or other countries outside Japan, such as Indonesia and other countries. But so we have to use local seeds, as can be seen in this figure: waste activated sludge, sewage sludge, sediment from pond, paddy soil, and others. So we have already performed the enrichment experiment in National University of Singapore and also UGM in Georgia. This is a photograph of the experiment for the enrichment of thermophilic mesanogens. Okay, as can be seen in this photograph, in NUS, we construct the handmade bioreactor like this. On the other hand, in UGA, photographed in the right side, the fermenter system is used. This is equipped with automatic feeding system of substrate, and the pH constant operation can be done in this system. This is the data in Singapore, but as can be seen in this figure, orange line is gas production. At the initial phase of the experiment, during 0 to 20 days of the duration, there are no gas production or slightly lower amount of biogas production. But this biogas production gradually increased, and the day of 60 days, biogas production became stable. So we concluded that 2-3 months is necessary to obtain thermophilic microorganisms. And this is the result of collaboration research with UGM. These figures show the gradual predominancy of thermophilic mesanogens. Relative abundance of some file function in the methanogenesis gradually increased while seed sludge was out from the reactor. And on the other hand, we applied quantitative PCR analysis. This revealed that predominant thermophilic methanogens, such as methansulfactor and methansulcina, propagated during the first 25 days of cultivation. Indonesia is the world's largest palm production. The plantation is 15.7 megahectare. The pome effluent from the palm oil extraction has great potential for biogas production. So in future, we'd like to deploy the thermophilic methanogenic technology, hopefully in Indonesia. And now we are collaborated with UGA. We have a 3-party agreement with UGM and Wika, which is the largest construction company of Indonesia, and Kajima. I am now supporting to Kajima to realize this technology. The picture showing my on-site instruction in UGM, you can see the UGM student and researcher gathering to the meeting. So this is my conclusion of talk. Mesogenic fermentation is decomposing reaction of organic compounds, methane, under anaerobic condition. The reactions were performed by a complex microbial system, which is called Microflora, carrying out seeds metabolism of electrons and metabolite in order to produce methane gas. Bioreactor is an apparatus that maximizes the reaction occurring in nature. The keyword of my talk is biogas, the Mesanogens, anaerobic digestion, biomass energy, and microflora. Also, please check my name or Kajima. You can see several papers. This time, I don't take time to introduce the detail of the technique or technology, but we have been published the data in some international journals. So I would appreciate if you could check these articles if you are interested in. This is acknowledgement. The Kajima Technical Research Institute, Dr. Tatara and Ms. Sumire Nakakoji, and Mr. Tsukada in the Singapore office of Kajima, and from UGM, Dr. Viratoni, Ms. Mariana Mati, and Lira, and Dani, and Mr. Rifki, and others. This is a photograph. Last time I visited to UGM, it was February last year, 2020, just before COVID-19 pandemic. Hopefully, I'd like to visit UGM again when coronavirus disappeared. I'm looking forward to seeing you all again. That's all of my presentation. Very sorry to inconvenience. Thank you very much.
Okay, thank you very much for the great explanation, Dr. Ueno. So we still have a little bit of time, about 2 minutes for the Q&A session. Do you have any question for the participant? We have 2 minutes left for the Q&A session. So you could send your question from the chat room or raise your hand. Once again, I welcome you to ask your question. Okay. Yosmina Tapilatu. Okay. Miss Yosmina Tapilatu, could you please unmute yourself? Okay.
Yes, thank you. Thank you for the opportunity to ask the question. Thank you, Dr. Ueno, for the very interesting presentation. I just have a short question. To obtain the seed, how long does it take for you to make the isolation? And if there is any particular media that is applied in the process? I ask you this because I work in the Center for Deep Sea Research, and it's interesting to know that there are also, I know that there are also methanogenic bacteria and or archaea in the deep sea, but it would be interesting also to do the attempt of isolating from the deep sea area as well, because I saw the last part of your presentation was about the isolation from the seeds were obtained from like paddy fields or the other area, but it's in a terrestrial area. So I was wondering if there is something also we can do concerning samples from the deep sea. Thank you. That's all my question.
Okay, very good question. Thank you very much. I can say if you want to isolate the pure strain of Mesanogen from natural environment, it is not easy to isolate because they are sensitive for the laboratory condition. So, but in our case, we enrich microflora, which is a mixed population of microorganisms, all of course contains Mesanogens. So live with others. They require the coexistence of other microorganisms because of various reasons there are. So nutrient and other reasons affect to grow the single microorganisms. So it is very difficult to isolate the single strain as a pure culture, but not so difficult to enrich the microflora as a microflora. But it is not, it might be not satisfied for the microbiologists because it is very difficult to identify whole microorganisms in the microflora. Is that okay? Can you hear
Miss
me?
Mina, is the answer to your content?
Yes, thank you very much. So what I can understand is that the seeds used here is actually like a microbial consortium. Is that it?
Yes, yes.
Okay.
Microbial consortium.
Mhm.
Yeah, and
Okay, thank you.
Yeah, enrichment of microbial consortia is not difficult.
Exactly. Okay.
Yes.
Okay, I think that's conclude the Q&A session because the time is already up. But in the chat room there is already another question directed to Dr. Ueno. So Dr. Ueno, if you have some time, could you please answer the question via the chat room?
Okay, I welcome. And also, please email me. I can answer as possible I can.
Okay, thank you very much, Dr. Ueno. I will send to the committee to share the email to
Thank
the
you.
participant. Okay, thank you very much for Dr. Ueno and the amazing explanation about the topics today. And let us give our biggest round of applause to him.
Thank you very much, Rifki. Stay safe and healthy.
Yeah, hope you and your family will stay safe during this pandemic. Okay, because the second session has come to an end, I would like to return the lead to the MC. Thank you very much, and sorry, I would like to apologize for any mistake that was made throughout the session. See you later.
Bye-bye.
As Master of Ceremony, I would like to thank Dr. Yoshiyuki Ueno for the lecture class today, as well as our moderator, Mr. Rifki Wahyu Kurnianto, Master of Engineering. Now we are heading to the photo session. As usual, I will do the countdown, and for the staff in charge, please be ready to take the pictures. To our dearest participants, please turn on your camera and show us your best smile. Now for the first slide. On the count of 3. 1, 2, 3, pose. Next for the second slide. On the count of 3. 1, 2, 3, pose. Next for the third slide. On the count of 3. 1, 2, 3, pose. Now for the fourth slide. And for the fifth slide. Next for the sixth slide. And last for the seventh slide. Alright, I feel like that's enough for the photo session.